Fertilization is the process in which the male gametes fuses with the female gametes to form a zygote, and in our topic, in most flowering plants, they have both male and female reproductive structures. It means that they are hermaphrodites. In the structure of flowering plants, you can see the female structure carpel, which consists stigma, style, ovary, and oval, and the male structure stamen, which consists filament and anther. So let's talk about the synthesis of the male gametes, which occur inside the anthers and called microsporogenesis. Inside the anthers, diploid pollen mother cells divide by meiosis to form 4 haploid cells. The nuclei of each of these haploid cells then divide by mitosis, but the cell itself doesn't divide. It means that cytokinesis doesn't take place, resulting in cells that each contain 2 haploid nuclei. These cells then mature into pollen grains, each surrounded by a protective pole made up of 2 layers. The tough outer layer called exine, which protects the grain from external factors, and the thinner inner layer called intine, which helps maintain the structure and function of the grain. One of the haploid nuclei is called the pollen tube nucleus, and the other is the generative nucleus. What are polar tube and generative cells used for? I will explain this later when we talk about fertilization. So these are the male gametes. Now let's talk about synthesis of female gametes, which called megasporogenesis. Inside each oval, a large diploid megaspore mother cell develops. This cell divides by meiosis to produce 4 haploid megaspore cells. The 3 cells are degenerated, and only one surviving haploid cell develops into an embryonic sac. The embryo sac grows larger and its haploid nucleus divides by mitosis 3 times, 1, 2, and 3, forming 8 haploid nuclei. As a result, we have mature embryo sac. One of these becomes the female gametes, 2 are polar nuclei, 2 are synergid, and 2 are antipodal cells. I have another picture here because we are missing some structural parts. The first is the filiform apparatus. It is formed during the development of synergids. Its function is to direct the growth of the pollen tube in the right direction, to provide transportation of nutrients to the embryonic sac. And another structure is the micropolar end and callosal end. Micropolar end is the entrance for the pollen tube where the ovum is located. And callosal end is the base of the ovule, connecting it to the plant and providing nutrition. And now let's talk about fertilization itself. First of all, we have a release of pollen grains from the anthers. Next, these pollen grains fall onto the female parts of the flower. And after pollen is deposited on the stigma, it must germinate and grow through the style to reach the ovule. The pollen contains 2 cells, the pollen tube cell and the generative cell. I was talking about them earlier. The pollen tube cell grows into a pollen tube through which the generative cell travels. The germination of the pollen tube requires water, oxygen, and certain chemical signals. As it travels through the style to reach the embryo sac, the pollen tube's growth is supported by the tissues of the style. During this process, if the generative cell hasn't already split into 2 cells, it now divides to form 2 sperm cells. And so let's enlarge the picture to see the interaction of the male gametes and the embryonic sac up close. In blue I have shown the embryonic sac, and here we see that the male gametes have reached it. I now zoom in on the embryo sac and we can see its structure again. In the beginning, without synergids, the pollen tube will not even reach the embryo sac because the pollen tube gets the direction with the help of them. Synergids release chemicals that guide the pollen tube, and filiform apparatus show the parts through which the pollen tube have to enter. One of the synergids degenerates, creating space for pollen tube's entrance, and one by one, both these male gametes will enter the cell. Now, after the release of the male gametes, this pollen tube has no role to play, and therefore it also disintegrates and disappears. Of the two sperm cells, one sperm fertilizes the egg cell, forming a diploid zygote. The other sperm fuses with the two polar nuclei, forming a triploid cell called the primary endosperm nucleus, which is a nutritive tissue that provides nourishment as the zygote develops. Since the male gametes fused twice, once to form the zygote and next to form the primary endosperm nucleus, this entire process is called double fertilization. After fertilization is complete, no other sperm can enter and the synergids with the antipodal disappear.